Morning everyone. Hi, I hope everyone is well and fit. We're starting teaching online, as you know, because of the coronavirus pandemic. So today we're gonna talk about the T cells and the T cell receptor with the importance of each of them, and I'm gonna talk about as well the MSC class 1 and MSC class 2 because they have an important role in activate the immune systems and how can the peptide fragments attach or connect with the T cells and activate the immune cells. Um, so at the beginning, I will let you know about the T cells functions, about how does the T cells work. So we are continuing our lectures with the T cells and immunology. Today is a very important lecture. We will be talking about the T cells functions. This is actually in the functions and the structures of the T cells. And we will continue on the B cells after this. T cells are a huge regulator of the immune systems, and there is a cross-linking between the innate arm of the immune systems and the acquired arm. So the T cells participate in amplifying the innate arm. In today's lecture, the things that we should be looking for are following number, and at the end of the lecture we should understand why and how various interactions between T cells and the other cells. For examples, the interactions between the macrophage, as I mentioned before, and I explained that what is the macrophage with the T cells. But today we need to understand what is the T cells exactly, and we should know as well the interactions between the T cells and another T cells. For examples, we've got the cytotoxic T cells, we've got the T helper T cells, so we need to go through each of them and understand what is each of them as well. So interactions as well we need between the T cells and B cells. You need to know as well the cytokines which is released from the B cells, such as the interleukin 2, interleukin 4, interleukin 5, and the interferon gamma, macrophage. They play an important role within the immune system. So they would actually go towards the functions of the T cells itself, and we want to understand how to T cells performs types of actions. So at the beginning, well, first of all, before we start the T cells function, as I said, we will need to know what's the T cells. So the T cells, as I mentioned here, belong to a group of the white blood cells called the lymphocytes. And white blood cells, as a general, protect the body from infections, and the main job of T cells is to fight infections. There are a number of the different types of the T cells, and I will explain that later, that act in a many ways to identify, attack, or destroy infections agents, because we've got like the intracellular pathogens and we've got extracellular pathogens, and we want to know which T cells will match with the extracellular antigen and which the T cells will match the intracellular or extracellular, so either extracellular or either endocellular pathogen. We want to understand it. Along with the other white blood cells, they play a major roles in the immune systems, which, well, basically it has an important role to enhance the immune systems. And we want to know as well, how does the T lymphocyte work? And the T lymphocyte, because as I mentioned, the T lymphocyte has different types of the cells. Um, so here we want to know the T cells come out from the thymus, and these are native T cells, which is called the naive T cells. So immature lymphocyte released from bone marrow. But the T cells matures in the thymus. But if you talk about the T cells, so the T cells come out from the thymus. But we've go to the immature and mature cells, because that means they, uh, well, the these T cells are a type of the white blood cells, as I mentioned, known as the lymphocytes. When the lymphocytes, as I told you before, it's to protect the body from the against pathogens and the cancer cells. The pathogens, it means like the could be bacteria, or it could be virus, or could be parasite, and the cancer cells as well. So because we know the pathways of the activate immune systems or enhance the immune cells, it has the different way to actually with the cancer cells or with another kind of the pathogens. So here in this slide, we concentrate about the lymphocyte exactly. In the lymphocytes, basically, we've especially the T cells, it's the immature, it's released from the bone marrow and but mature in the thymus. But the B cells mature already in the bone marrow, so release and mature in the bone marrow. The thymus is an organ located in upper chest, and the immature lymphocytes leave the bone marrow and then find their way to thymus, where they are educated to become mature lymphocyte. So T cells lymphocyte develops from the stem cells in the bone marrow, and these immature T cells migrate to the thymus via the blood, and the thymus is a lymphatic system gland and functions mainly to promote the development of mature T cells. In fact, the T cell in T cell lymphocyte stand for the thymus drovii. T cells lymphocytes are necessary for the cell-mediated immunity, which is an immune response that involves the activation of the immune cells to fight infections, and the T cells functions to actively destroy infected cells, as well as to signal other immune cells to participate in the immune response. Okay, here we've got two types of lymphocytes. We've got the B lymphocytes, which is the activated humoral immunity and release antibodies, and we've got the T lymphocyte, which is non-antibody producing cells, which they activated the mediated arm of immunity. T cells are lymphocyte immune cells that protect the body from the pathogens and the cancer cells, and the T cells originated from bone marrow and mature in thymus. They are important for the cell-mediated immunity and the activation of the immune cells to fight infections. So the T cells, it's divided into many types. The first one, which is the T cytotoxic T cells, actively destroyed infected cells through the use of the granule sacs that contain digestive enzymes. So basically the T cytotoxic T cells is kill the cell straight away by releasing the granules. These granules, it has ability to induce the apoptosis. As you know, apoptosis is the cell program death. We've got the T helper cells as well. T helper cells, because its name's help cells, that's mean to activate the cytotoxic T cells and boost the immune systems, or call the other immune cells to help the T cytotoxic to help the macrophage to stimulate the antibody productions by the B cells lymphocyte. In reality, we've got the regulatory T cells suppress the actions of the B cells and T cells to decrease the immune response when a highly active response is no longer risk or that's the dangerous. So regulatory T cells, that mean it's to regulate the cytokines storms, because you know when the T cells activate the immune systems, we need to something to stop this. So the immune systems, it's just we wanted to know that how it works, like the highly active response on the immune systems, and without causes any risk or to disrupt the immune cells. And we've got as well the natural killer cells or natural T cells, with that they distinguish the infected cells, especially the virus or the cancerous cells, from the normal body cells and attack the cells. Do not contain the molecular markers that identify them as body cells. We've got as well the memory cells protect against previously encountered antigens or may provide the lifetime protections against some pathogens. Well, the T cells and the B lymphocytes are a central player in adaptive immune systems, and they have ability to generate the highly specific cell surface receptors. Both the T cells and the B cells they have receptors or different receptors on the cell surface. These receptors will basically co-receptors on their cell surface that bind the MSC molecules and provide additional strength to the bond between the T cells and the target T cells. And then through the genetics recombinations of a relatively limited number of a precursor for these receptors, the T cell receptors, and the B cell receptor as well. So the T cell is called the T cell receptors, but the B cells receptors called antibodies, and can be generated that are specific for the particular molecular structures called antigens. And the antigens, it could be different from the sites, could be the proteins, could be amino acid or the glycoproteins, and they come from the bacteria or parasite or virus, or could be the cancer. And then basically we know the T cell receptors, it's very important because there are a protein complex founded on the surface of the B cells or the T cells, and is responsible for recognizing fragments of antigen as a peptide bound to MSC molecules, and each locus can produce a variety of polypeptides with constant and variable regions. I will explain that later with the details, the compounds of the T cell receptors. But the B cells it will be the next lessons. Here we've got as well, as I mentioned before, we've got many types of the T cells, but we've got two measures importance type of the cells. We've cytotoxic T cells and the helper T cells. Cytotoxic T cells, it's going to kill certain cells, but the helper cells is trying to help the T cytotoxic. And we've got the T suppressor, T memory cells, and we've got the originated, it differentiated from the stem cells and sensitized the T lymphocyte produce a clonal of the T cells, have different functions. We've got as well natural killer cells. Although the lymphocyte play a major role within the innate immune response, but these cells are it has it can call a police that presents of the special antigen presenting molecule that are expressed on the, all cells in the body, and they play a key role or an important role in the operations of the adaptive immune system. Natural killer cells use the special receptors expressed on the cell surface, and this is the receptors called the natural killer receptors. And the name is different because it's used the germline encoded receptors. So it could be like it's activated receptor or inhibitor receptors. Basically, it's that distinguish. It could be the receptor is different actually from the T cells of the B cells, and has ability to kill the cells that express abnormal MSC receptor profile as well as the signs of infection. So the natural killer cells or natural killer cells is a lymphocyte able to bind to the certain tumor cells or and virus infected cells without the stimulation of the antigens, and kill them by the insertions of the granules containing the GRAN enzymes or peripherin. So that's, well, it could be the same enzymes releases from the T cells because it's a part of the T cells as I mentioned. So the natural killer cells are the granular cells that containing tiny vesicles that store a powerful digestive protein such as the peripherin or GRAN enzymes. They bind to cancer or infected cells. They release this protein which causes the abnormal cells to lyse. When the cells, for instance, display little or no MSC protein on their membrane, this triggers the natural killer cells to bind to destroy the cells. Okay, so basically in here I mentioned okay, here the natural killer cells, here the target cells, natural killer cells. It's a kind of the T lymphocytes release the granules toxins, which is called the peripherin or GRAN enzymes. That is the release. So it's activated or lead to lyse the cells without no MSC protein. So there is no MSC protein. This can trigger the natural killer cell to bind to the destroyed cells. So in here, natural killer cells can kill target cells by two measures mechanisms: the death receptors, because basically the natural killer cells can target wide variety of the cell types, and natural killer cells looks for that the abnormal cells, like I mentioned. This could be the viral infections or it could be the cancer cells. And when the cells infected with the virus, okay, with the virus, for instance, that cells filled expressions of the self MSC class 1, and the natural killer cell looked for proteins on the surface of the proteins and able to bind and activation receptors of the natural killer cells. Natural killer cells, it has the receptor which is called the death receptors depending killing. These receptors will recognize there is something antigen or on the expressed on the target cells that will lead to activated or to release some cytotoxic granules. These granules, it has the digestives, like digestives granules lead to release some protease which is form the pore inside the the cells, sorry about that, and then lead to activation of the natural killer cells, leads to polarizations of the granules, and within that then either make the mitochondrial release like the cytochrome C's and then into the cytoplasm. After that will lead it to reduce the apoptosis and then trigger the pathway of the apoptosis, because maybe you've known about the intrinsic pathway and extrinsic pathway of the apoptosis, which means the cell death program. So basically, now you go to that the natural killer cells is like the basically is a granules cells which is contain some vesicles. These vesicles, it has digestive proteins such as the perforin and gran enzymes, which is when they release, they lead to distract the cells and damage the whole cells. Basically, the natural killer cells, it could be the ask it that lead to the it's a part of the innate immunity, and well, yeah, is a part, that's right, but the normal cells basically that's contain normal antigen, which is self antigen and MSC class 1, but sometimes in the cancer cell they lose the proper sequences of nucleotide to create proper proteins known as the MSC class 1. Okay, and then you go to the whole idea about the natural killer cells, and there I will explain the MSC class 1 and MSC class 2, so you get the further information about each of them. Okay, so the structures of the T cell receptors, as I mentioned before, the T cell receptor, it's expressed the some glycoproteins or some the fragments proteins on the cell surface. So they need these small proteins, or the fragments peptide, they need to bind to the T cell receptors, and then can they activated the immune systems. So basically, the rule of the T cell receptors in the immune systems, well, basically is just to like to engage them and activated the immune response between the innate immunity and adaptive immunity, like because they express it on the T cells, and the T cells is a part of the adaptive immunity, and the fragments proteins, it expressed on the macrophage or dendritic cells, or it could be the B cells as well, and the B cells is a part of adaptive immunity, but that wouldn't mind. But that means it's the cross link between the innate immunity and adaptive immunity. So we want here to understand the structures of the T cell receptors. Basically, the structures we want to know the T cell receptor. T cell receptors, if you look here, this is the T cell, okay? Here the T cells, and we've got here the receptors of the T cells. Okay, so we've got two kinds of the receptors. It's look like the immunoglobulin or the IgG or the IG. Okay, I'm not going to say IgG or IgA or just IG as a structure. So but there was the differences between each of them, like the name and but the like the functions, or it could be the structures, it's the same because it has the variable constants transmembrane. Okay, so here the T cell receptor and here the T cells. Let's then say the T cell receptor. Okay, a T cell receptor is a protein complex found on the cell surface of the T cell. This is the T cell and this is the T cell receptor, and the receptor is a protein complex. Okay, and T lymphocyte that is responsible for recognizing the fragments antigen, because if you look into this groove here on the T cell receptor, okay, here the antigens, okay, the fragments antigens complex that's bound to the measure histocompatibility on the other side here, and I will explain that later as well. That will sit on this groove, okay, that will lead to send the signal to the T cells, and then they release the cytokines which is lead to activated the immune systems. So the binding between the T cell receptors and the antigen peptide is of relatively low affinity and is degenerated. That is, the many T cell receptor recognize the same antigen peptide, and many antigen peptide recognized by T cell receptor. So let's describe the T cell receptor. Basically, we've got here, this is the T cell receptor, is composed of two different protein chain. That is the heterodimer. In a human, we've got 90% of the T cells receptor consists of the alpha change and the beta change. So 90% of the T cell receptor is alpha and beta change, whereas in the 5% of the T cells, that's the T cell receptor, is basically is a gamma and delta T cells. Okay, so this is the gamma and delta T cells, and we basically, so there are two types of the T cells that can be distinguished by the T cell receptors. One of them it's called the alpha beta receptor. It's contained the T cell receptor as alpha and beta subunit, and while the gamma and delta T cells, these have the receptor consisting of the gamma and delta subunit and T lymphocytes. So this is a part of the cell-mediated immunity. So as I said before, it's look like the IgG, the IG, sorry, I said the IgG, no, it's wrong, IG, because we cannot say it's IgG or IgA, but I'm talking about the structures, you know, the structures of the of the immunoglobulin for IgG or IgA, it's different. So we need to just to be more precise or accurate when you talk about the structures of the T cells. Basically, we've got here the both alpha and beta change. These alpha and beta change, we've got, this is, it's called the transmembrane region, or this here, this area, it's a transmembrane regions, which is in the T cells. The gamma, alpha and beta change of the T cell receptor, it has the cytotoxic cell. This is cytotoxic cell that will help to, when the antigen, okay, it comes here and bind to this receptor on the T cells, okay, T cell receptor, that will send the signal to inside the cells through the cytotoxic cell and will lead to activated the signaling pathway inside the T cells. Okay, this is the variable region, which is called the V, and this is the constant region, that's for both, I mean for the alpha and beta T cells and the gamma delta T cells. So we've got the variable regions, constant regions, and the cytoplasmic cell. The variable region, constant regions, it looks like the IG, immunoglobulin, both of them. The alpha change, beta change in 90 or 95% of the T cells, but the gamma and delta T cells is about just 5% of the T cells. Okay, so this is the structures of the T cell receptors. As I mentioned, it has the cytotoxic cells, which is send the signals when it comes from the antigen to inside the T cells. Here, the T cells, here the types of the T cells. We've got alpha, beta change, okay, gamma, delta chain. Okay, we've got the variable region, we've got the constant region, we've got two types of the T cells. Okay, cool. So I'm going to talk about here a measure histocompatibility complex. A measure histocompatibility complex, it could be, or which is the MSC class 1. So the antigens as you know, it could be the any kind of the pathogens, bacteria or the parasite or the virus, or it could be cancers, so induce the immune response inside the body. And most of these antigens are large proteins molecules. Some are polysaccharide and a few are glycoprotein or nucleoprotein. So we also came to know about the self-antigens and the autoantigens. So the antigens which are found on the membrane of almost all the cells of the human body and vertebrates are known as the MSC, okay, as the MSC antigens. There are glycoprotein in, so the MSC is glycoprotein in chemical nature. So MSC class 1 or MSC, discovery of the MSC antigen when the scientists were doing the transplantation experiment on the mice. Scientists found that sometimes that the transplant tissue from the donor mouse was expected by the recipient mouse, but at the other times it was rejected by the recipient mouse. Why the tissue from the one individual of species were destroyed when they introduced it into members of the same species? The answer was given by an American mouse genetics called the George Snell. He gave the reason. Rejection was because the tissue of the donor and the recipient were incompatible. That is mean the MSC class 1 wasn't incompatible with the recipient. In other words, if the animals are not closely related, the recipient rejected the graft and its mean in genetics background. So I will talk about the transplantation later and about the MSC class 1 or MSC. Let's say the measure histocompatibility molecules incompatible. So MS or measure histocompatibility or MSC class molecules is a set of gene. Okay, this is a set of gene located on the chromosome 6 of the human. There are a linkage of the set of the genetic loci encoding protein. Okay, and the encoding proteins involved in antigen process and the presentation to the T cells. The measure histocompatibility class plays an important role in immune response. I will explain that later in each part. MSC molecules are, so if you want to know, the MSC molecules are a peptide are delivered to the cell surface by the MSC molecules. T cells recognize the peptide MSC complex. So I explain that later because we want to understand some important point about how can the MSC or the peptide, the MSC class 1 or MSC class 2, and it has the, well, it holds the like the peptide fragments on the cell surface and will react with the T cells and the T cell receptors can recognize them. So we need to more details and more information about it to understand it, the important points about the measure histocompatibility. So the important thing we want to know, the MSC class 1 is a peptide MSC complex, and it has the two types of the molecules. One is MSC class 1 and MSC class 2. There is the groove here, which is the peptide fragments will insert inside this groove. We've go to them. Each class presents peptide antigen of the two different type of cells, CD4 and CD8. CD4 for two helper cells, CD8 for the T cytotoxic cell. Okay, please focus on these points okay, because they are very important to know about the MSC class 1. Firstly, the MSC class 1 is a set of the genes located on the chromosome 6, and the MSC are a peptide, okay, our peptides are delivered to the cell surface by the MSC class 1 and the MSC class 2. The MSC class 1 expressed on the CD8, okay, on the CD8. MSC class 2 expressed, okay, oh sorry, it's not expressed, it means bind. The MSC class 1 um, well, bind to the CD8 receptor, T cell receptor CD8. It means the CD cytotoxic, T cell cytotoxic. And the class 2, which is the MSC class 2, will bind to the CD4, which is T helper cells. Okay, so the main functions of the MSC molecule is to bring antigen to the cell surface by recognized by the T cells. Okay, so I will explain that. Here we've got the cell, any kind of cells. It could be our cells or it could be any cells of the immune systems. Okay, the cells express, it has the MSC. It could be MSC class 1 or it could be MSC class 2. Here the antigens, because when the antigen invade the cells, that will lead to express it on the MSC. It could be MSC class 1 or it could be MSC class 2, and I will explain that later. Okay, and then after that, here will the T cell receptors will bind to these fragments peptides, okay, and then can recognize what? Then can recognize there is an foreign body, okay, invade the body and will enhance the immune systems. Okay, so let's understand some important points about measure histocompatibility complex. So measure histocompatibility, or the histo, it means tissue. Compatibility means getting along or agreeable. MSC got it. It's complex. So these genes are localized to the large genetic regions, contains multiple loci, and a tightly linked cluster of the genes whose products play an important role in intracellular recognitions and in discrimination between self and non-self antigens presented on the chromosome 6. We've got here the two type of the MSC molecules. We've got MSC class 2. We've got the MSC class 1. MSC class 2 molecules are also transmembrane glycoprotein. So the MSC class 1 or MSC class 2, sorry, it's a transmembrane protein in the cases consisting of the alpha and beta polypeptide change of the molecular weight 34 kilodalton and 29 kilodalton respectively. So if we want to define the MSC class 2, so MSC class 2 is transmembrane glycoproteins expressed on the professional antigen presenting cells, such as the dendritic cells, the neutrophil or the B cells, and the macrophage. Okay, so the MSC class 2 is just expressed on the dendritic cells, B cells, and the macrophage. And it's the transmembrane glycoprotein in these cases consisting of the alpha and beta polypeptide changes of the molecular weight 34 kilodalton and 29 kilodalton. There are considerable sequencing homology with the class 1, and the structure studies have shown that the alpha 2 and beta 2 domains, okay, and the ones nearest to the cell membrane assume that the characteristic Ig folds, as I mentioned, so it's the Ig like the immunoglobulin, while the alpha 1 and beta 1 domains mimic the class 1 and class 1, alpha 1 and alpha 2 in forming in a groove or bounding by 2 alpha helix and the alpha beta pleated sheet flow. So I will explain that in detail in the next diagram about the MSC class 1 and MSC class 2. This is like the general information about the structures of the MSC class 2 and the MSC class 1. So the MSC class 1 is founded on it's not like the MSC class 2. MSC class 1, and please concentrate on what I'm saying, okay? MSC class 1 is focused or founded on the all nucleated cells in the body and they play important role in humoral and cellular immunity. Class 1 molecules consist of the heavy polypeptide chain of 44 kilodalton and non-covalently linked to the similar 12 kilodalton polypeptide called the beta 2 microglobulin. So the differences between the MSC class 1 and the MSC class 2 is the MSC class 2 is found or exists on the professional antigen presenting cells, which are dendritic cells and macrophage and the B cells. But the MSC class 1 is founded in all nucleated cells. Okay, the structures of the MSC class 1, it consists of the alpha and beta 2 immunoglobulin or polypeptide, but the MSC class 2 consisting of the alpha and beta polypeptide chains, and the molecular weight for them it was a heavy polypeptide chain which is consists of the 45 kilodalton in MSC class 1, but in MSC class 2 the molecular weight is 35 and the 29 kilodalton, and here no it's linked to similar basically the class 1 molecules consist of the heavy polypeptide chain of 45 kilodalton and linked to the smaller 12 kilodalton polypeptide called the beta 2 microglobulin. So the largest part of the heavy chain is recognized in 2-3 globular domain. Well, basically I know it's a bit hard and it's a bit complicated, it's tricky, so you need to whiteboard to explain the whole details about the MSC class 1 and MSC class 2, but sorry, I will do that in the classroom, so don't worry if you don't understand any words I'm saying, so you can just email me or you can write the private comments. I will explain that, and when I will see you on the 30th of May, they will in the classroom I will explain every details what you want. So just to read it and get some information about it, and then we can have a chat about all of them and we can go through. So the basically the globulars and the 2-3 globulars domains alpha 1 and alpha 2 alpha 3, that sequences it carries in the C terminals into the cytoplasm, and the solution of the crystal structures of the human class 1 molecules provided an exciting leap forward in our understanding in functionals. So both the beta 2 microglobulin and the alpha 3 regions resemble classical IG domains. IG domains mean the immunoglobulin, so it looks like the IG domains in folding group? Well, because they are, it looks like the receptor expressed on the cell surface, like the T cell receptor. However, the alpha 1 and the alpha 2 domains, which are most distal to the membrane, form to extend the alpha helicals above a flower created by strands held together in the beta sheet. The whole forming is wall forming in the groove of the MSC class 1. So basically, I know it's a bit tricky, but I'm trying to explain what I wrote in here. So just we've go to the diagram, the next slide, about the MSC class 1 and MSC class 2, and then we will explain it. Okay. Here, this is the diagram about the MSC class 1 and MSC class 2. Diagram showing domains and the transmembrane segments of class 1 and and MSC class 2 molecules. So, well, basically, this is the structures of the MSC class 1. I will start it with the MSC class 1. Okay. So basically, the MSC class 1 expressed on the nucleated cells and consists of the two polypeptide chains. Okay, two polypeptide chains. How? It's two polypeptide chain. This is its alpha chain. This is the beta microglobulin. So first thing, MSC class 1 expressed on nucleated cells consists of the two polypeptide chains. Alpha, okay, this is the first polypeptide chain. This is the second one, which is called the beta microglobulin. Okay, the beta microglobulin. Alpha chain, okay, and beta microglobulin. Alpha chain, okay, has three external domains, okay, has three external domains, which is called the alpha 1, okay, alpha 2, and the alpha 3. But it has a short, well, basically the alpha 1, and it has a short cytoplasmic tail, alpha 3. Okay, which one? The alpha 3, attached to the plasma membrane, and alpha 3 bound to beta microglobulin, bounded non-covalently to the alpha chain. Essential for proper folding of the alpha chain, it has no transmembrane region. Okay, it has no transmembrane region. Identical in the all cells present intracellular or endogenous antigen on the surface, present the peptide on the groove. Okay, so we need to focus and on this diagram of the MSC. This is the MSC class 1. This is, okay, the MSC class 1. MSC class 1 expressed on the membrane of the all nucleated cells, on the membrane of the all nucleated cells, is consist from the two polypeptide chains. The one, it's the alpha 1, the alpha chain, and the second one, it's the beta microglobulin. Okay, the alpha chain consists of the alpha 1, alpha 2, and alpha 3. The alpha 3, it has a short which is called the cytoplasmic tail, and attached to the plasma membrane of the nucleated cells. The alpha 3 attached to the microglobulin and the beta 2 microglobulin. Beta 2 microglobulin it's bound to the alpha 3 non-covalently. Okay, so if you go back to your and it has these, that mean it's the C region or C terminus, that is mean that will lead to stay the MSC class 2 inside the cell, sorry. Here we've got the glyph or the groove of the MSC class 2 that will lead to release when the antigen. Well, in MSC class 1, you have to know the they express the virus and the cancer on the cell surface. Okay, the cancer, so endogenous microorganisms or endogenous pathogens. The endogenous pathogens is mean the virus or means the cancer. Okay, so that will express on the cell surface of the cells, and the antigens will display here on the groove of the MSC class 1. This is which is called the alpha helix, okay, in the alpha chain polypeptide, and we've got this one which is called the beta pleated sheet. Okay, so now we'll jump to the class MSC class 2. Okay, the structures of the MSC class 2. MSC class 2 basically presented on what? So we need to know the differences between each other. Here, so I will explain that later when we get done from the diagram of the MSC class 2. So the presented on the antigen presenting cells, the expressed MSC class 2 molecules on the professional antigen presenting cells, B cells, macrophage, and the dendritic cells. So the first thing, MSC class 2 express on the dendritic cells, on the B cells, and the macrophage, okay, which is called the professional antigen presenting cells. The MSC class 2, it's could present it or express it for the short periods on the which is called the non-professional antigen presenting cells, such as the microglia in the brain or the fibroblast, but is much more restricted, so it's not the express it for the long time, it's just for the short time. Okay, so the MSC class 2 consists of the two molecules, okay, consists of the two molecules. The molecules alpha and beta, okay, alpha and beta chain. Both are in core in the plasma membrane, so both are inserted in a plasma membrane. Okay, it has two external domain, which is called the alpha 1 and the alpha 2, okay, and beta chain, okay, beta 1 and beta 2. It consists of the two external domains as well, beta 1 and beta chain. So the MSC class 1, it consists of the alpha and beta chain, okay. This is the alpha 1 and alpha 2, beta 1 and beta 2. So the beta 1 consists of the two external domains, beta 1 and beta 2. As I mentioned, beta 2 domain contains the binding site of the T cell co-receptors, okay? And also the alpha 1 alpha 1 and beta 1 domains from the antigen binding pocket. present extracellular or exogenous antigen on the surface of the MSC class 2, such as bacteria, and they break down the peptide fragments are presented on the surface of the peptide groove. Sorry. So we've got here, this is the MSC class 2. MSC class 2 expressed on the nucleated of the membrane antigen presenting or professional antigen presenting cells such as B cells, dendritic cells, and the macrophage. It has two molecules, alpha change and beta change. Alpha change has two external domain, when alpha 1 and alpha 2, and beta 1 and the beta, it has two external domains called beta 1 and beta 2. Both of them are inserted or embedded in the cytoplasmic or in the membrane of the cells. Okay, and they have the basically it has the groove, okay, in the upper of the MSC class 2. These here, well, the bacteria or the antigen will present on the surface of the peptide binding groove, okay, on the peptide binding groove. So the peptide fragment. So that means here it's the present extracellular or exogenous antigen, which is bacteria or parasite. Okay, so if I want to compare between the MSC class 1 and the MSC class 2, okay, let's start it. This is the MSC class 1 and MSC class 2. MSC class 1 expressed on the all nucleated membrane. MSC class 2 expressed on the professional antigen presenting cells such as dendritic cells, macrophage, and the B cell. Okay, the structures of the MSC class 1 consist of the alpha change, 2 polypeptide change, alpha change, and the beta microglobulin. Okay, but the MSC class 2 is consisted from the alpha and the beta change. Okay, the alpha change polypeptide in MSC class 1 consists of the alpha 1, alpha 2, and alpha 3, with the beta microglobulin, and the alpha 3 bound to non-covalently with the beta microglobulin. Okay, the alpha 3, it has the cytoplasmic cell inserted in the membrane of the cells. Okay, that will lead to stay the MSC class 1 inside the cells. In the MSC class 2, we've got the alpha 1 and alpha 2, that means two external chains, and the beta 1 and the beta 2, both of them are embedded in the cytoplasmic membrane of the cells, and because it has the cytoplasmic cell. The MSC class 1 presented the exogenous antigens, which is the bacteria, or which could be the parasite. But the MSC class 1, which presented just the cancer or the endogenous molecules like the cancer or the virus. Okay, so that's the diagrams showing the domains. So you know the domains, you know the transmembrane segments of the MSC class 1 and MSC class 2. So you know the structures of both of them. Then we're gonna talk about what's going on inside each of them. Okay, so the cross presentation of extracellular antigens on the MSC class 1 molecules by the dendritic cells. Basically, the dendritic cells is a part of the innate immune response. Okay, so and this is the dendritic cells will bind to the T cells. So that means the dendritic cells or the macrophage or the neutrophil is across all its like the participants of the innate arm with the innate adaptive arm. So there's a cross linking between each other. And we've got the many kinds of the pathogens. We've got like the exogenous antigens and we've got the endogenous antigens. The exogenous antigens basically the endogenous antigens that's expressed on the MSC class 1. So maybe they will ask you why you say it here the cross representation of exogenous antigens. That's mean the exogenous mean that when they express it on the cell surface of the MSC class 1 will be exogenous. Endogenous it's mean inside. I'm talking about the antigens. I'm not talking about when the pathogens or the fragment peptides express it on the on the dendritic cells or the antigen presenting cells. So the certain subsets of the dendritic cells are efficient in capturing exogenous protein and loading peptide divide from them onto MSC class 2. So basically the first line defense of the immune system is the dendritic cells. The dendritic cells is a part of the leukocyte, okay, and I mentioned that before. So it's circulated in our body and then it gets mature, and when they get matured then will be like the dendritic cells, like these arms. That is means it's used to engulf the pathogens. So when they engulf the pathogen, the pathogen then will go through inside the cells, and when go through inside the cell then will present it after the long process in the cytoplasm, in the cytosols, then will degrade for degradations by you know about the protosome. I think you've heard about that. It's like the panel shape inside the cytoplasm or the cytosol. It has like the digestive enzymes and with the high acidity to break down the microorganisms into many peptides. That will lead, and then they send the microorganisms to the endoplasmic reticulum. When the endoplasmic reticulum, there are some chaperones will take the peptide then to the cell surface of the antigen presenting cells, which is could be the here it's the MSC class 1, and the usual and another route may involve direct transport of the antigen from the phagolysosome into vesicles, loading the peptide fragments, and where the peptides are allowed to be bound to the mature MSC class 1 molecules. Basically, the MSC class 1 is expressed in all nucleated cells. It's not on the just like the MSC class 2, it's just on the antigen professional or professional antigen presenting cells. No, but there are the special process. How can the microorganisms gets into the cells and then express it on the MSC class 1? Now you understand the T cells, okay? It came from the famous and then matured, get matured. When get matured, they go to the T cell receptor on the cell surface. The T cell receptor is should recognize with the MSC class 1, which is expressed on the some kind uh, of the innate immune cells, like the dendritic cells or the macrophage or the neutrophils. And then after that will activate it, lead the signals to the inside the T cells, and when they send the signal to inside the T cell, that will lead to release some cytokines and then lead to destroy the foreign body. Okay, so I'm gonna talk about the basically about the how can the pathogens get into the cells. Okay, so here we go to, this is the diagrams at the beginning. This is the our cells. Okay, you've got the inside your cells, the nucleus. Okay, it should be here. This is the endoplasmic reticulum. This is the Golgi apparatus. This is the process and will happen in the inside the cells. Okay. This is the MSC class 1. MSC class 1, as I told you, it has the cytoplasmic cell, which is say the signals inside to the cells. Okay, this is the peptide fragments, will they express it on the groove of the MSC class 1, and this is here the T cell and expressed the T cell receptor on the cell surface. So we want to understand how the peptide fragments display on the MSC class 1 and how interact with the T cells and lead to activated the T cell through the T cell receptor. Okay, so the T cells and the B cell recognize different types of antigens. B cells, can recognize variety of the antigen. The antigens could be, as I mentioned, protein, polypeptide, nucleic acid, and lipids. But the T cell recognize only antigens peptide. MSC class 1 presents intracellular or endogenous antigen, such as virus or the cancer. Intracellular pathogens live and replicate inside the cell's host, like the pandemic coronavirus. So it's invade the nucleated cells, and it should be the MSC class 1 expressed on the MSC class 1 and lead to activated um, the CD8 or this the CD cytotoxic T cells. But basically in the coronavirus, we've got something which is called the cytokine storm. That will happen and lead to disrupt the immune systems and cannot work properly. So this is not our topic, but I give you just an example about that, just to get into. Okay, so basically, and under the normal condition, the peptide or the proteins will basically derive from the cytosol, or any nucleated cells are displayed by the MSC class 1 and the peptide. When a cell infected by an intracellular pathogens, for example, um, virus or cancer, MSC class 1 molecules present the peptide derived from the virus for recognition by the T cells, and the MSC class 2 expressed the extracellular pathogens live and replicated outside host cells. So extracellular bacteria, or toxin or parasites, So basically, let's talk about the MSC class 1 and how can present the peptide fragments on the cell surface of the MSC class 1. Okay, so the MSC, well, the breakdown of the antigen into fragments peptide of this pathogen is presented by the MSC for the recognition by the T cells. The process by which pathogens, their products, are degraded to produce peptide antigen is known as antigen processing. Okay, so what does mean antigen processing? This process by which pathogens and their products are degraded to produce peptide antigen. Okay, these peptide fragments combine with the MSC class 1 molecules inside the cells. How? It's because the cells, it has the, like, it's is because the MSC class 1 is formed inside the endoplasmic reticulum, and the virus or the cancer, it's replicated inside the cells that will carry the MSC class 1, the pathogens or the peptide fragments with them. Okay, uh, and then express it on the cell surface of the MSC class 1. The MSC peptide complex, this formed travels from the cell surface, where it displays peptide fragments to the T cells and now in the antigen presenting cells. So okay, so the MSC peptide complex, this forms travels from the inside the cells where display the peptide fragments to the T cells, which is called now the antigen presentation. Okay, I will repeat that. The process by which the pathogens on their products are degraded to produce peptide antigens is called antigen processing. These peptide fragments combine with the MSC molecules inside the cells called the antigen processing. When the MSC, okay, peptide complex formed travel to the cell surface and carry or displays peptide fragments, okay, to the T cells, this is known as the antigen presenting cell. Okay, the antigen presenting cells. Well, so we want to know how peptide fragments pathogens and their products are produced, and how these peptide fragments are combined with the MSC molecules, and how MSC peptide complex is presented to the T cells, processing and presentations of the intracellular endogenous antigens cells. So basically we've got this is the cells of the human body, and the cells, it has like the nucleus could be here, and this is the endoplasmic reticulum, this is the Golgi apparatus, this is the proteasome. Okay, I will take an example of virus. Okay, a cell infected with virus. Okay, let's say for instance coronavirus or any kind of virus, because it's the pandemic right now. Okay, and when the virus infected the cells, okay, is the virus, we know the virus when gets into inside the cells, that's will there is a cellular machinery of the cells to synthesize the viral protein, and this occurs in the cytoplasm of the target cells. Okay, there is a cellular machinery inside the cytoplasm, which is called the cellular machinery to synthesis what? To synthesis the viral protein. That's mean it's makes a viral like segments or small pieces. And when the virus infected the cells, um, this occur in the cytoplasm of the where of the target cells. This is our cells, for instance. Okay, this is our cells. The virus get in. There is a cellular machinery inside the cells. The cellular machinery will uses will cellular machinery of the cells to synthesize what? The viral protein. And the digestive in the large spiral shape, which is called the proteasome. Proteasome, viral protein are taking and digested, okay? Are what? Are digested in the proteasome. Proteasome, the environment of the proteasome is acidic. It's highly acidity, and that will lead to breakdown of the made of about 12 to 15 protein subunits, because the proteasome have a proteolytic activity, and this they are degraded the cytosolic protein into peptide fragments. Okay, look at this, the peptide fragments, okay, are about 8 to 5 amino acid long. Okay, and then so the viral protein are digested by the proteasome. Okay, by proteasome, and these peptide transported to the rough endoplasmic reticulum. Okay, to the rough endoplasmic reticulum. This transportation occurs through a pore formed through a pore. Okay, these pore formed by a protein, now a tap, which is transported associated with antigen processing. TAP is consists from two subunits, TAP1 and TAP2, and alpha change with beta change, and then of the MSC class 1, are synthesized in the endoplasmic reticulum and then translocated to the end of these change are assembled together. Okay, when assembled together near the TAP site, and then the one peptide have entered to endoplasmic reticulum by the MSC class 1 in the peptide binding groove. Then the MSC release from the chaperones, there is some chaperones, release from the chaperone to the cell surface via the Golgi apparatus. Okay, so the cell surface integrate into membrane and recognize it by the T cells. So T cell receptor can recognize the peptide presented on the MSC class 1 molecule, and it also has the co-receptors of the CD8, which is recognized, and the alpha 3 domains of the MSC class. The T cells interact with the both peptide MSC class 1 molecules on the cell surface of the target cells. Okay, I know it's a bit complicated and it's a bit tricky, but I'm going to report it repeat it, sorry, the whole points. So basically, you know, the MSC class 1 is expressed on the nucleated cells. So we've got, for example, the viral when it gets inside the cells, that will lead, there is the machinery proteins, or cellular machinery that will lead degrade the viral into small pieces. When the virus gets into the proteasome, that will lead it to to made of 12 to 15 protein subunits, because the proteasome has the proteolytic activities. This is the peptides will get to the endoplasmic reticulum. The endoplasmic reticulum has the two has the TAP, which is the peptide transported protein. This transported protein, which is called the transported associated with the antigens, that the TAP consists of the two subunit, TAP1 and TAP2, the alpha domain and the beta change of the MSC molecule art synthesis, and the translocated to endoplasmic reticulum. So basically these changes are assembled together with because the MSC class 1 as well is mated or assembled in endoplasmic reticulums comes with the chaperones. Then the fragments of proteins will bind with the MSC class 2 and then express it, and then can go and express it on the cell surface of the on the nucleated cells, through the Golgi apparatus. There's the T cell receptors will bind it with the peptide, and this is the T cell, which is basically the C cytotoxic, because the MSC class 1 can recognize the CD or the kind bind with the CD8, as I mentioned before, with the T cell. We've got and the co-receptors, the co-receptors that can recognize the alpha 2 domains, alpha 3 domains of the MSC class 1. So this is the basically the pathway of the MSC class 1 when the peptide of the microorganisms gets inside the cells, and then express or exist on the cell surface of the nucleated cells. Basically, let's jump to the MSC. So this is the presentation of the cellular antigens by the MSC class 2 molecules. So the MSC class 2 molecules, well, as I mentioned before, express it on the antigen presenting cells such as the dendritic cells or the B cells or the macrophage. And basically the MSC class 2 pathway can deliver the cytosolic antigen for presentation by the MSC class 2 on the cell surface. It could be the macrophage or it could be the dendritic cells. So the portion this comes out from of the cytosols taking in autophagosomes, because we've got the phagosomes inside the cytoplasm fused with the endocytic vessels and with the lysosomes. That will lead it to distracted. This is the peptide, and then can the MSC class 1 carry and express it on the cell surface of the immature dendritic cells and expresses the peptide, and then can the basically the T cells, which is the T helper cells, can recognize this is pathogens. So the MSC class 1 can bind with can recognize it by the T cells cytotoxic, but the CD4 it could be recognized by the MSC class 2 can be recognized by the T helper cells, and both that will lead to activated their immune systems, because the MSC class 1 it's founded in all nucleated cells, and then it has the new presented the endogenous pathogens like the cancer and the virus. So that will lead straight away to goes to the CD8 can recognize it straight away and release some cytokines and the granules enzymes and lead to destroy it and call the macrophage to engulf them. But in the MSC class 2, so the pathway they can the pathway it could be different. So how we can the microorganisms get in, because it comes from the outside of the cells like the bacteria or the parasites. We've got vagosomes will fuse it with the lysosome. That will lead it to after that will lead to distracted the pathogens into peptide fragments, then express it on the MSC class 2. So I will give them more details about the how can the pathogens get in the MSc class 2. So well, I'm sorry about these yeah about this lecture. Really I know it's a bit complicated for you, but I'm trying to explain. This is your curriculum, so I made it more easier for you, but I hope you can understand it each point. So please, if you have any questions about any part of the session, please let me know in the private comments, or you can email me, and I will explain that to you. And maybe when we go back to the campus, so we can in the classroom explain the whole details about, and if you know further informations about the topic, so I will let you know. So please let me know. Okay, so then we'll jump to the explain how they present the cellular antigens by the MSc class 2, and then can the T cells recognize. So please, well, just well, just focus on this slide to understand it. The MSc class 2 presented the cellular antigens. So the antigens that originated from the outside host cells, extracellular bacteria. These antigens are taken inside by the cell either by endocytosis or phagocytosis. The cells involved in this pathway are antigen presenting cells, which is called antigen presenting cells. And the pathway starts when the extracellular antigens, okay, extracellular antigens, basically such as the extracellular bacteria, are translocated by the antigen presenting cells. Antigen presenting cells enter listed pathogens into intracellular vesicles, now the endosome. Okay, so endosome, and the endosome basically fused with the lysosome, okay, inside the cells. So this is like the bacteria or any extracellular microorganisms will get into the cells. In the pathway, it's different to the MSc class 1. So you wanted to know the antigen presenting cells internalize a pathogen into intracellular vesicles known as the endosome. In the next step, okay, the endosome fused with the lysosome. The lysosome, which is called the endolysosome. Endolysosome internalized pathogens are digested by the proteolytic lysosome enzyme and into small peptide fragments. Okay, so internalized pathogens are digested by proteolytic lysosomes enzymes into small peptide fragments. The MSc class 2 molecules are synthesized and assembled where? In endoplasmic reticulum, like the MSc class 2, okay? Class 1, and have the beta change of the molecules of the MSc class 1 is associated with the third protein change called the invariant chain, okay, the invariant change, and then bind to the grooves of the MSc class 1. This is a binding stop self protein in the left endoplasmic reticulum from bind to MSc. So invariant MSc class 2 are transported from the endoplasmic reticulum to the Golgi apparatus, from the Golgi apparatus to another vesicles, which is called the environment. So only small fragments peptides which bounded to the groove of the MSc class 2, and the fragments which is known is called the clip, okay, and this associated invariable change polypeptide. These vesicles contain the MSc class 2 and bound to the clip fused to the vesicles containing the peptide fragments internalized antigen. The peptide fragments display the clip and binding to the peptide binding group. Okay, to the peptide binding group are 11 to 15 amino acids and long, and then newly formed the MSC peptide complex and is transported to the cell surface. Once the cell surface are presented to the T cells, which is that lead, which the T cells, it's the T cells which is the CD4 T cells, and the T cell can recognize it, the peptide, and display on the MSC class 2 molecules by the T cell receptor, which is the co-receptor known as a CD4 binding to beta 2 domains of the MSC class 2 molecules. So I'm sorry about that, it's a bit hard and complicated, but I will repeat it. Okay, so here the pathogens, and this is the protein, like the exogenous pathogens will get inside the cells, and inside the cells we've got the endosome. The endosome will fuse it with the lysosome, and then will break down the peptide or the exogenous or the pathogens into small fragments peptides. And then we've got here in cross response the endoplasmic reticulum, which is made the MSC class 1. The MSC class 1 will okay, so assemble in the rough endoplasmic reticulum, and then there is the MSC class 1, which is basically it's moved from, and then there is through the and then the peptide or the small peptide will bind to the cell surface, to the group of the MSC class 1 that's mated in the Golgi apparatus. There is some structures which is called the fragments, which is called the clip. It's founded on the surface of the MSC class 2. This is the vesicles contains basically the MSC class 2 molecules bound to the clip, and the clip fused with the vesicles containing the peptide. Okay, so these vesicles contain what? Contain the MSC class 1 and the MSC class 2, and bound it to what? To bound it to the clip. Okay, and these and fuse it with the vesicles. Okay, and the vesicles, it has what? It has the peptide fragments of the antigens. Okay, and then the peptide fragment display the clip and binding to the peptide binding groove. Okay, the peptide binding groove, and then formed the and then transported to the cell surface, and then display of the MSC class 2 and the core. There is then the CD4 can recognize the T cells, and well, basically the can recognize this is the fragments peptide which is expressed on the MSC class 2, and then the co-receptor as well of the T cell will bind it to the MSC class 2. Okay, so this is about the MSC class 1 and MSC class 2, and you've got the whole information about each other. So we will continue with the activation of the T cells, C cytotoxic, because it's very important and it helps. So, okay, the C cytotoxic or the CD8. So now you've learned about the T cell receptor, about the MSC class 1, MSC class 2, how the pathogens can get in, how many types we've got about the T cells. So we've got the two measures types of the T cells, which is the T cytotoxic and the T helper cells, and we've talked about as well the natural killer cells. But now we want to know the cytotoxic T cells. So basically the CD8 plus T cells become cytotoxic, and the cytotoxic T cells kill the infected cells. By doing so, they eliminate the reservoir of infections. These cells are known as T killer cells. T lymphocyte abbreviation, well, to the the CD8 it could be, or T cell, and basically they directly kill the cells that display the foreign antigens on the surface of what? Of MSC class 1. The main functions of the cytotoxic T cells is to kill the cells that have been infected by the virus or could be the cancer. They are also involved in killing of tumor cells and the transplanted foreign cells. So let's see how the cytotoxic T cell work and when intracellular infected, for example, viral infections and the dendritic cells from the site of the infected migrated to nearby the lymph node and presented the viral antigen in the form of the MSC1 more complex. The naive T cells, CD8 positive or T cells, it's bound to the antigens at the same time, never activated the T helper cells. Basically, the T cytotoxics, when attached to other cells of the body, if the cytotoxic T cell learns that the cells is infected or the pathogenic, so it will do two things. The first thing, it's force the cells to commit suicide, which is mean the apoptosis, which is a programmed cell death, or secreted the granular enzymes, which is called, that will kill the cells straight away. So basically in here we want to know about the principal mechanisms of the cytotoxic T cells. Basically that the actions is the calcium dependent release of the cytoplasmic. The calcium dependent release, that will when the calcium gets in, that will lead it to activated to release some cytokines, which is called, I will explain the cytokines in the next lesson of each of them, and you want to know about each of them. So that will release the granules and recognition the antigens on the surface cytotoxic. The cytotoxic granules are modified the lysosomes that contain at least the three classes of the cytotoxic effector proteins, which is the cytotoxic effector protein, which is called the perforin. We've got the granenzyme and granulosin. These protein, the three proteins that will release from the cytotoxic T cells, and that will lead to destroy the infected or the target cells. So basically the perforin, for instance, act by forming the pores. Okay, when the forming the pores, or like the make a big hole inside the cells, the target cells, the plasma membrane, which both causes the direct damage to the target cells and the forms the will lead to another like the gran enzymes gets in the sites and then destroyed the cells. We've got the last one, which is called the granulysin, which is expressed in the human but not in the mice, has the antimicrobial activities, is high concentration, also able to induce the apoptosis and necrosis. Well, the gran enzymes activate the apoptosis once delivered to the target cells of the cytosols and via pores and by perforin and the cytotoxic granules, which act as the sac-fold forming complex with perforin and the gran enzymes. That means in this slide, basically we've go to the T cells, and the T cell, with the when the binding to the cells, it's expressed, there's some pathogens on the MSC class 1, the T cells will release some cytotoxic granules. These cytotoxic granules will penetrate the cells and induce it, well, basically it's made the holes or pores inside the cells that will lead it to induces the apoptosis and effect lead to damage of the DNA and induce the program cell death. Then that's the one the well called the macrophage, or the another kind of the cells just to engulf the distracted cells or the target cells. Okay. So here the effector cytotoxic T cells secreted some cytokines and become itself stimulated. And when stimulated, these cytotoxic T cells leave the lymph node and migrated to the site of infections to attack virally infected cells. So all cells expressed the MSC class 1. This is all the infected cells which expressed the MSC class 1 on the cell surface, which is under the normal conditions to display the host proteins, which could be the self-antigen or non-self antigens. It means the self-antigen which is found inside the body, and basically the non-self antigens, it means like the virus, or there could be the cancer. And then they express it when they're infected, and then display the foreign antigen, therefore in this case the viral antigens. Okay, so display on the infected cells at the MSC class 1 molecules complex. The effective on the D cells recognize it and bind it to the MSC class 1 through the T cell receptor, which is kill the cytotoxic T cells. And then the cytotoxic T cells make direct contact with and it's mediated by the T cell receptor and co-receptor CD8. The T cell receptors bind to antigen display by the MSC molecules. Okay, cytotoxic T cells undergo degranulations, and so the granules of the cytotoxic T cells migrated to the site of the contacts of the migrate contact and release. The cytotoxic T cells contain a specific cytotoxic which is performed cytotoxic effector such as the perforin and gran enzymes. Both of the gran enzyme, or these are located in the granules of the inside the CD3, and they are loaded into granules, and perforin forms the pore inside the cells or the target cells and form passage the granules, and the commits into the target cells, and then induces the apoptosis and ingested by the phagocytes. That's what the pathway of the CT cytotoxic T cells. So we've got here, this is the macro, the infected cells, which is it could be carried the foreign antigen or the self antigens, which is the self antigens could be in our body, or it could be that it's non-self antigens, could be the pathogens like the cancer or the virus. That's we've got the T cell receptor here. The T cell receptors is bound to the CD8 T cells. The T cells has the granules enzymes which are called the perforin and the gran enzymes. These, when the T cell receptors will bind to the peptide fragments on the MSC class 1, that will lead to release the perforin and the gran enzymes and make the pore inside the cells. That will lead to induce the apoptosis, and then ingested by the phagocytes will come the other cells to engulf these infected cells. This is the pathway of the CD cytotoxic when they infected with any foreign bodies, and what will gonna happen. Okay, so basically now you know how to express the pathogens on the MSC class 1, okay, which could be the on the nucleated cells, and we've got the T cells as well, and you know the T cells as well. It has the T cell receptor and how the fragments peptides bound to the T cell receptor. So, okay, so we've got here the T helper T cells, which is the CD4 T cells. T cells basically assist the other leukocyte in immunological process, including maturation of the B cells into plasma cells, basically, and the memory T cells, and activated the cytotoxic T cells and the macrophage cells. So T helper cell, which is called express it, the marker, it's called the CD4, but the CD cytotoxic is the CD8. That is very important to know. The T helper cells are activated by interactions between the interactions between the T cell receptors and the peptide, as I mentioned, it's the same process with the measure histocompatibility type 2, which is expressed on the surface of the antigen presenting cells. And then the process rapidly then lead to proliferate the T helper cells and the subtypes that will lead to secreted a lot of the cytokines, and that will lead to activated the immune cells. So the T helper cells, it works as a booster to immune system or trigger to immune system, or to help the others kind of the cells when for instance the CD8 binding to the pathogens that will the CD or the helper T cells will come to activate the immune system called the other kinds of the cells to help the T cells receptor. Okay, and then the get the signaling pattern from the antigen presenting cells. Okay, so basically they will, when the T helper cells activated, it will divided into several subtypes, which is called T helper 1, T helper 2, T helper 3, T helper 17. So we've got the many subtypes created from the basically from the immune cells. And I will show you the pictures of the T helper cells, how it's activated. So this is the activation of the T cells. The T helper cells, you know, it's do not directly kill the infected cells as the cytotoxic, and basically, as I mentioned, it's once activated, they divide rapidly, regulating the adaptive immune systems by signaling from the antigen presenting cells to the direct differentiator of the T cells into many subtypes, which could be the T helper 1, T helper 2, 3, 7, T or 9, which is depending on the required immune response. It's created different cytokines to facilitate immunoglobulin class switching, because the T helper cells as well will activate it as well the B lymphocyte. It's not just the T site, it's help the T cytotoxic, but what we call the B lymphocytes, and facilitated with the productions of the enhance the productions of the macrophage through direct interactions and release the soluble factors, which is the targeted intracellular pathogens such as the bacteria or parasite via activation of the macrophage. Okay, so we want to know the one of the important things which is called the T cells activations. We want, you know, how the peptide fragments express it on the antigen presenting cells, if it's macrophage or dendritic cells, or it could be B cells, and this is how interact with the T cells. It could be T helper cells, or it could be what? It could be T cytotoxic cells. Okay, so basically, when the mature dendritic cells is a critical step in the cell-mediated immunity, the T lymphocytes occurs in where? In the bone marrow, as I mentioned before. All the cell migrated to the thymus to complete their maturation. So the maturities of the T cells then found circulated in the lymph node. Most T cells are considered naive or immature, CD8 or CD4, have a potential to become cytotoxic or helper. Okay, then the pathogen different pathogen via presentations of their antigens on the cell surface of migrated dendritic cells mature. Once the lymph node and the antigenic cargo of the dendritic cells presented by the MSC class 1 is recognized by naive T cells, undergo the activation period. How? So the recognitions happen, the first recognition or first activation of the T cells, the number one is happens through when the T cells can recognize the T cell receptor, okay, of the T helper cells, or it could be T cytotoxic T cells, recognize the peptide fragments on the T cell receptor. Okay, the T cell expresses another marker which is co-stimulatory, its interaction with another marker of the CD of the dendritic cells to synthesize the which is the interleukin, I mean the cytokines here. And then the interleukin on the dendritic cell with the professional and differentiated of the cells, then ultimately lead to the clonal expansions of the another kind of the T cells, and then they lead to the clonal and the surface of the of the CD8 or the CD4 is the surface of the glycoprotein, which is a primer expressed on the cytotoxic T cells and act a co-receptor for the T cell receptor. So basically, you've got here the dendritic cells, okay? So I will explain that. Dendritic cells, and we've got here the MSC class 2 express like the peptide fragments. We've got here the T cell, T helper cells, and we've got the T cytotoxic T cells. The T helper cells, when bind to the MSC class 2, or bind to the peptide expressed on MSC class 2, that will lead to enhance the cytotoxic T cell to or activated the cell mediate immunity attacked on infected cells, and the same times will that lead to activated the humoral immunity to secreted a B cell to secreted a more plasma cells. So I will go through each point of the T cells activation in the next. So basically here the T cells activation through the 3-signal pathway. Okay, what is the 3-signal pathway? At the 3 signals, once the lymph node and we've got here the dendritic cells, which is the antigen presenting cells. This is the MSC class 1. This is MSC class 1, it could be or MSC class 2. And here the peptide fragments, here the T cell receptor, and here the T cells. The first interactions when the T cell receptor it's binded to the peptide fragments on the MSC class 1. Okay, and the second interactions when the CD8 bind to the or interact with the co-stimulatory molecules, which is called the B7, which is the CD8 and CD86 on the dendritic cells, or it could be the antigen presenting any antigen presenting cells, lead then the third signal, which is the lead to release the interactions of the interleukin 12 and 2, that will lead to enhance the T cells to release the interleukin 2, and then that will lead it to the that's mean the T cells activation. So the T cells activation, it's happened through the three signals events. The number one, which is when the T cell receptor bind to the peptide fragments on the MSC class 1. The second one, when the CD22 expressed on the T cells and bind to the B7, which is the CD80 and the CD86 marker on the antigen presenting cell, that will enhance to release the interleukin 12, which lead to release the interleukin to activate it to release the interleukin 2 in the T cells, which is called the cytotoxic signaling. So that the T cell signaling pathway, we've got co-stimulatory interactions, we've got the cytokines signaling pathway. Okay, well, I'm going to stop here. I know this lesson could be complicated and tricky, but please, if you've got any questions or any any inquiry about the lessons, you can just email me, or you can just write private comments to me, and I can just reply you as soon as possible, or maybe I will explain anything you don't understand it in the classroom. Thank you very much for your listening, and keep safe. Okay, bye.